Okay, thanks very much, and welcome to this initial session actually at the Vancouver Public Library. We're very pleased to be down here, and so thank you all very much for coming. I think you're in for a bit of a treat tonight. I'm just going to say a couple of words about the Life Sciences Institute before we begin. First of all, this is, of course, a partnership, the Vancouver Public Library and the Irving K. Barber Learning Centre of UBC. Is putting this on tonight. But moving on to the next slide, just say a couple of words about the Life Sciences Institute at UBC. It's one of UBC's major institutes. There's about approximately 900 people and 900 scientists in that building. UBC is ranked #1 in Canada in life science research, number 23 in the world according to international rankings. And so it's the Life Sciences Institute as the lead, the Life Sciences Institute at UBC is really the anchor for that endeavor. So it's a huge research endeavor. We bring in around about $40 million a year in research funding, and we have ambitions to do much better than that. So there's, as I mentioned, there's about 90 laboratories. Sometimes the number goes between 900 and 1,100 researchers, somewhat. Let's just say the place is really big, 500,000 square feet, etc. And the Life Sciences Institute, in addition to doing basic research, which is, of course, the fundamental mandate, and it's the area where the new findings that will have great impact on human health will come from. But in addition to that, it's really initiating a much closer contact with the healthcare and the clinical community through a personalized medicine approach. And the personalized medicine is really the medicine of the future, where each of us gets our genome sequenced, their proteins analyzed in our blood, our microbiome analyzed, essentially getting a huge data cloud around all of our cells that will be very predictive for disorders that we may have and disorders we may get. And so the ambition here is to be able to prevent disease well before it actually occurs. And this is, as I say, something that I think all of us have as a real ambition within the center. So personalized medicine is, as I mentioned, an individualized approach to healthcare. All of us are different from each other in substantial ways. Drugs that work on one of us don't work on the other one. And so we need to analyze what our particular molecular makeup in order to be able to treat whatever disorders one has in the most effective way. So we're going to have much better ways to predict, diagnose, and treat disease. Very much improved healthcare delivery, where we're able to diagnose much more accurately what an individual has, and then match that more precisely to the disease, to the treatment that is going to be given. Improved efficiency, only use the drugs that actually work on people, and then reduce the number of adverse drug reactions. Right now, adverse drug reactions are about the 5th leading cause of death in North America. For people over 65, are usually on about five or so medications. The potential for adverse drug reactions is huge, and about 90% of adverse drug reactions aren't reported. So this gives you some idea of the scale of the problem. That's usually due to genetic factors that are different between one individual and another. And so there's a lot of reasons, a lot of very compelling reasons why the Life Sciences Institute is focusing in on this more individualized approach to healthcare. So tonight, what you're going to hear are some details of that more molecularly based approach, and it's to do with the immune system. If you think your brain is complicated, the immune system is even more complicated because without an immune system, we wouldn't live at all. We'd be eaten by whatever bug was around, and so our immune system is really the first thing that allows us to survive. And so what you're going to hear about tonight is really some of the leading people internationally who are going to give you a taste of what the immune system is all about and how we can manipulate it to live improved lives. The title here, I have to admit, is pretty good. Harnessing the immune system to treat infection. autoimmune diseases and cancer. So you're basically some of the major disorders of our time. So I'm going to hand it over now to Neelan Abraham, who's a professor in the Department of Microbiology and Immunology, and going to moderate this session.
Good evening, everybody. It's a delight for us to be here today. My name is Dr. Neelan Abraham. I'm a faculty member at the UBC Department of Microbiology and Immunology. So before we start, I'm just going to give you a bit of the lay of the land. I will introduce our speakers shortly, and thereafter we will be hearing from them, and I'm just going to lay out the program. We would like the speakers to have about 10 minutes each to speak. If any questions arise in your mind, I encourage you to use the paper and pens that are in the middle of the tables at each of your respective tables. So you would write your questions on them, and then during the course of the forum, I or Stephanie Butlin, who's our LSI outreach coordinator standing there waving a hand, will collect those questions from you, and we will then take a short break where we'd like you to actually mingle, go get yourself some refreshments, discuss questions that come up, and get your questions into me. Then we will reconvene, and I will ask those questions and direct them to the panel members who will then address them. Just a housekeeping note, there is a concern about whether you're being recorded. Only the speakers are being recorded on video and audio, and so the cameras are directed at the speakers, not at the audience. Your questions will be read by me, and that will be recorded as well, and then none of your images will be used publicly in any way or form. So let me introduce our speakers. The first speaker will be Dr. Georgia Perona-Wright, and then that'll be followed by Dr. Ken Harder, who's sitting next to her, and finally we will close out with Dr. Mark Horwitz, and then after their talks we will then welcome you to mingle, write your questions down, and then we will take it from there. Thank you.
Brilliant. Well, thanks very much. Thanks all of you for coming. I think this evening is meant to be an evening where we enjoy some immunology. And my job here was to provide a somewhat simple introduction to it, so you can see how well I get on with that. I wanted to start with a photo just to show you the beauty of immunology. And the particular cell type that I'm showing you here is a macrophage, and I'm only using that word because it has a really interesting root. If you follow the Greek, the macrophage is a big eater. So it's a cell after my own heart, and the reason to start was just to illustrate the interaction there. To illustrate the interaction of the immune system with some bacteria here, ready to be eaten by this macrophage. But before I really get going, I wanted to ask a couple of questions of you. I know it's a mixed audience tonight. How many of you have actually studied immunology? I know there are some familiar faces, so there's a fair few. Brilliant. How many of you have ever had an allergic reaction? So a fair few more. How many of you have ever had going the wrong way? Ever known anyone or yourselves been affected by a transplantation, organ failure and transplantation, or anyone affected by cancer? So these are all interactions with the immune system. And of course, the other one is infection. So I'd say, how many of you have had an infection? I'm sure that is the whole room. How many of you have ever sat on the bus or sat in a library lecture next to somebody that's sneezing? These are all interactions with the immune system. So this is a sneeze-in process, and you should think about that when you're close to somebody on the bus. So this is the immune system, and I wanted to describe a little bit about what it actually is. And so this was my best image of the immune system. This is exactly what the immune system looks like. It is also known as Cardiff Castle in Wales. And what I wanted to do here was to draw out some of the aspects that are similar between the physical defenses that we know in army terms and the immune defenses that we have in our own bodies. So the immune system is a defensive system. It's what is protecting us against invasion by different bugs, by different pathogens. And so there are a few kind of key similarities there. And the first one is the very structural thing. So if you're designing a defense system, one thing to do is to prioritize where you place your defenses. So the fact that there are very limited entries to this, to the castle here is very similar with us, that our body is enclosed, our skin and so on provide a structural defense against any invasion. And more than that, when you think about the different layers of structures we have, it's not just the obvious ones you see, but it's some on the inside as well. So not only our skin, but also the mucus, the saliva in our mouth, the mucus that lines our gut, the snot in our nose. All of these are structural defenses that are designed to make it more difficult for pathogens to get into our body. And so those are part of the immune system. And then there's a very fluid part of the immune system, and those are the individual cells and molecules that make up our internal immune systems. And if you were to continue to draw the parallels to the castle here, these would be our soldiers, our lookout guards, the soldiers that are posted in order to sense an invasion or to spot an invasion before it gets close. And then the foot soldiers, who are the ones whose job it is to actually tackle that invader and take it down. And those foot soldiers use a variety of different weapons, and we'll talk about those in a minute. But there are also some aspects here that you don't immediately see when you look at the castle from the outside. And those are the commanding officers, or the central command, and of course those are sheltered inside in a very protected space. But those are actually the important guys, because those are the ones that are making the decisions. Those are the ones that are choosing which particular aspect of this defense system to use in any particular context. And the other really important part of an effective army is the training, the education and the training that will ensure that that army is most effective in its action. And so if you really want to get rid of a particular invasion, what you want to do is to work out what its Achilles heel is, work out what its weakness is, and learn to exploit that. And so the first interaction with an invader enables you to learn a little bit about it, and that means that the second time you see that particular invader, you can deal with that in a much better way. And that memory, or that process of education, is a really important part of the immune system. And we'll come to that towards the end of the talk. So if this is what the key points of immunology are in castle terms, this is what they are in immune system terms. So what I'm really talking about are the different cells and the different molecules that are involved in the immune system and how those all interact together. And I just wanted to introduce you to a few of the key players, because I think Ken and Mark will talk about some of these later. But these are some of the cells, some of the soldiers, some of the effector arms of the immune system that are really important for getting rid of those pathogens. So my particular favorite is the T cell here. There are also cells called B cells. And both of these are, well, it depends who you talk to, but I can talk about them as the foot soldiers of the immune system. These are the cells that actually go and do the dirty business of getting rid of that invading pathogen. And then there are some of the key commanding cells, and those are the dendritic cells. These can act as sentry guards, so they can act as the commanding officers, and they're the ones that control the type of immune response you get. So all these different cells will communicate with each other, interact with each other. And you know that the cells are critically important to immune response, because the recruitment of cells, or the accumulation of cells, is what is behind those swollen lymph nodes that tell you you've got an infection going on. So this poor lass is a lass with a serious throat infection, and you can tell that she's got a throat infection because she has massively swollen lymph nodes in her neck. And so you can see they're swelling even under her mouth here, in her neck, and around the bottom of her neck here. And these lymph nodes, these swollen glands, are just accumulations of lymphocytes. They're accumulations of immune cells that are getting ready to fight that infection. And so the cells really matter. The way they interact with each other also matters. And so some of the key molecules that we will talk about in the immune system are proteins, other molecules that cells will make, that they will spit out or release from the cell, and that will provide instructions to target cells. So let me tell you a little bit about my pictures here. So here we've got a T cell. Remember, the T cells are one of those foot soldiers of the immune system. And the business that this T cell is involved in here is killing a target cell. And the reason it wants to kill that other cell is because that cell is infected with a virus. And the easiest way to kill the virus is to kill the whole cell that contains it. And the way that it's killing it is by releasing these toxic granules, these little packets of toxic molecules, and the T cell is spitting them out on that target cell. And the outcome of that interaction will be death of this target cell. So the particular molecules that cells make can really determine the outcome of an immune response. And there's another type of molecule I wanted to introduce you to, and I think you'll have heard of this one. So these are antibodies. Antibodies are a product of B cells, the other foot soldiers in the immune system. And antibodies are things of some beauty. they have this really specific Y-shaped structure. And what the antibodies do is that they bind to very specific targets that are parts of the pathogen. They're parts of that invading microbe that is trying to mount the infection. And you can see this beautiful Y-shaped structure. If you look at, this is a photograph of the mucus from somebody's lungs. So you can see that these antibodies actually hang out in those in the sites that are likely to interact with pathogens. And they're there in order to help the immune system work. They're one of the effector molecules of the immune system. And the other point that I wanted to make about the immune system is that it's an incredible network. Like, part of the beauty of the immune system is the way that different cells interact with each other in a very fluid way. It's unlike any other system in the body in that it has to patrol the whole of your body and to do so in quite a mobile way. And so these cells will physically interact with each other in quite an intimate way. So this is the cells you're familiar with now, a dendritic cell interacting with a T cell. And you can see that they're so close that their membranes will physically align with each other. They will zip together. And so the cell-to-cell interaction is quite exquisite. So is the kind of whole organ interaction. And so what we're looking at here is all the dark shading. The purple shading here is staining of individual lymphocytes, individual immune cells. And they are lining up along the gut wall. So this is the lumen of somebody's gut. And this is the kind of tough tissue of the gut wall. And you can see that all of these cells are arranging themselves in a very particular structure that will enable them to fight off the infection that's coming in through that gut lumen. So the interaction between those cells and the communication between those cells is really quite exquisite. And that's some of the work that the three of us are doing, some of the research that the three of us are doing just now. So that is the immune system. That's all you need to know about the immune system. I think the question for tonight was, how can we actually use that immune system for our own benefit? Like, how can we exploit and manipulate that immune system in order to design new treatments and new therapies for a variety of different diseases? So what I wanted to think about first was the ways that we can use those tools of the immune system, those products of the immune system, to help us out in the situation of infection. And I thought there were three different ways that the immune system can be used to help us in infection. The first way is diagnosis, and then the second way is for treatment, and the last way is to try and prevent infection altogether. And so I'm going to go through those in 3 slides, and you can see what you think to those. So the first consideration was diagnosis. How is the immune system actually useful to us in diagnosing different diseases? And one of the really important molecules here is the antibodies that I was talking to you before. And the reason those antibodies are important molecules is that they are long-lived and that they last a long time, even after an infection has happened and has healed. And so they're like the smoking gun that is left behind, and they're a trail that will tell us that an immune response has happened. And so the beauty of that is that the antibodies are very specific, and they will tell us the particular pathogen or particular microbe that we have been infected with. So if you have a teenager that's tired and unwell and you want to find out whether she or he has mono, what you would do is go for a blood test. And what that blood test really is that it's testing that blood to see if there are antibodies against the virus that causes the mono. And so those antibodies will only be left behind if there has been an infection and the antibodies have been made. So they're a very clear indicator that an infection has occurred. And so we use that in a number of settings. A number of blood tests are antibody, or we're trying to detect antibodies. So the other classic example there is an HIV test. So if I am working in the lab with human blood and I foolishly stab myself with a needle, and I now need to wait and see if I've been infected with HIV from that needle stick, what we actually do is draw blood and look to see if there are antibodies in there specific for HIV. And that would be the indication that I have indeed had a HIV virus in my body. And so a lot of these tests are all reliant on the action and the trace of having an immune response. So that's one way we can use antibodies, is to pick up particular infections that have happened. But we've even learnt to exploit those antibodies to a different extent. And so we can create our own antibodies with known specificity, and we can use those in completely separate tests. So a lot of blood tests that you have, whether it's to monitor hormone levels, they are using specific antibodies that are designer antibodies made to pick up that hormone. And so a lot of our blood tests require exploitation of immune molecules. So testing for hormones is a great example of that. A pregnancy test is in fact antibodies on a stick. That's what you're using to test whether your urine has the right hormone in it. So we use antibodies in a huge number of ways, just as tools, as designer tools, to help us out, both in the clinic and also in the lab. And one of the examples in the lab, this is not my own research, but a group in Montana. But the reason, or the way that antibodies have been used here, is that the antibodies have been tied to different colored dyes. And by tying them to dyes means that we can see them on microscopes. And so when we put these antibodies onto particular cells, they will only bind if they're specific for that cell type. So it enables us to see things that we wouldn't be able to see in any other way. And so the science behind these, the pictures on the right here, are that the red is an antibody against macrophages. Do you remember macrophages, the big eaters? So the red is showing you the macrophages, the immune cells. And the green dye is tied to a different antibody, and that antibody is specific for Francisella, which is a bacteria. It's actually a really nasty bacteria. It causes tularemia, which has, it depends on the strain, but it can be up to an 80% fatality rate. So it's not a bacteria that you want to have around. And so this up here is using two different antibodies to show us the presence of bacteria in somebody's macrophages. And the really nice thing about this paper, where the pictures came from, is that they were testing out a new antibiotic. And you can see how effective that antibiotic is, because when those macrophages were exposed to the antibiotic, there is no bacteria left, right? All the bacteria have gone. So the tool here is the antibodies. The research is into antibiotics, but the antibodies have enabled us to read out this result. So that was using tools, using parts of the immune system for diagnosis, for diagnostic reasons. The next step is actually how to treat diseases. And so in thinking about infection, infection I have already spoken about in terms of an army trying to prevent invasion. And you can think about the army releasing its weaponry on the particular pathogen. But the pathogens are not stupid either, and they have ways of dealing with the immune system, finding ways to escape the immune system. And so we often talk about the immune response to infection as an arms race, that the immune response is desperately trying to quench the pathogen, just at the same time as the pathogen is trying to evolve ways to get around the weapons of the immune system. And most of the current treatments that we have for infections are treatments that try to disable the pathogen. So antibiotics, for instance, they can work in a variety of ways, but they will break up the cell wall of a bacteria, or they will stop a bacteria dividing. And by slowing down the ability of that bacteria to replicate, it gives the immune system a greater chance to actually defeat that infection and to clear it. So by using agents that stop the pathogen, you're hoping that the immune system will win out. And the only problem with that approach is that, as I said, the pathogens are pretty fast adapters. And if you target a weakness in a bacteria, the chances that that bacteria will evolve or will mutate in order to develop into a form that doesn't have that particular weakness. And that's the basis of antibiotic resistance. If we use antibiotics too frequently, we're encouraging the bacteria to develop a resistance to that, to develop into a form that doesn't need it. And so the alternative approach in this arms race is, instead of tackling the pathogen, actually just to try and help our own systems to overcome that infection. And by boosting the person's immune system, you're not providing the same opportunity for the bacteria to evolve around the particular weakness that you're focusing on. And so the idea is that in the era where antibiotics are becoming less and less useful because resistance is growing, that actually treatments that are aimed to enhance our own immune responses might be the solution to that. They might be able to plug the hole that is being left behind by antibiotic resistance. And so different ways, a lot of people are putting a lot of effort into designing different ways of enhancing the immune system there. Some of those are actually not new ideas. So the idea of providing anti-venom after being bitten by a snake. My foreigner geography tells me that this is the only venomous snake in British Columbia. I should be aware when I'm hiking. So if you are bitten by a snake and you need an instant response, what you actually need is an anti-venom. And that is an antibody against the venom. It's a way of enhancing your immune system. And so these ideas of helping our own defenses are not new ones. What is new is that we're thinking of more and more different ways to try and do that, different molecules that we can provide and different ways to enhance our own immunity. And so hepatitis C, chronic hepatitis C, is a lovely example of this. So hepatitis C is a wily virus, and most people who are infected become, it's not actually true, but a large proportion of people who are infected become chronically infected with hepatitis C. And what that means is that their immune system haven't been able to fight it off, and the virus settles in for a long ride. And so if you can actually boost the immune system of those people, and one way is to provide interferon, one of the immune molecules, that will enable or just kind of shift the balance between pathogen and host in favor of the host, in favor of the person. So that interferon therapy, the enhancement of the immune system there, is often good enough to clear the hepatitis virus. And so what I and what a lot of other people are working on just now is to try and push those ideas into other infections and to try and work out new ways of enhancing our own immune defenses. So there's a lot of potential in using the immune system to treat infection, and particularly in an era where antibiotics are becoming less useful. So that is diagnosis and treatment. And the last point that I wanted to mention briefly was prevention. So even better than diagnosing and treating people who are sick, the idea would be to prevent the infection taking hold in the first place. And the best way that we have to achieve that is through vaccination. And vaccination is purely an interaction with the immune system. So do you remember when we were talking about the castle? We spoke about education and training as an important part of generating an effective army. Vaccination is designed to supply that process of education. So the idea of vaccinating is that you have shown the pathogen to the person, enabled that person's immune system to have a go at fighting that pathogen, such that when that person encounters the same pathogen again, they're going to be much better at it. Their immune system is going to be faster, it's going to be stronger, and it's going to be much more tailored. So it's already going to have worked out the weakness of that particular pathogen, and having done that, it means that the type of immune response that is mounted the second time is a much better one. So the idea of vaccines is providing just enough of the pathogen that you have set off this process of training and education in the immune system. The key thing there is that you don't actually want to provide exactly the same pathogen, because the whole reason that you're vaccinating is that that pathogen might kill you. That's probably suboptimal in the vaccination process. And so vaccines they're not the whole pathogen. They're often killed pathogens. So we have heat killed them, or we have inactivated them in some way. They are sometimes just parts of pathogens, just single molecules or multiple molecules taken from the pathogen. And the other group, the group of live vaccines, is attenuated pathogens. So pathogens that are very similar, but are much less likely to make you sick. But similar enough that the immune response you mount against it is still going to protect you against the wild pathogen, the dangerous pathogen, should you encounter that one again. So that's the theory of vaccination. It's a brilliant way of preventing disease, and we know that because of some of the epidemiological data associated with it. So over on the left here, this is a child with smallpox. Smallpox was a really scary disease. It had an immensely high fatality rate, particularly in children. And you know that this photograph was taken in the early 1970s. And the reason that you know that is that the process of vaccination, which started in the 60s, completely eradicated smallpox from the globe. So after 1975, there is no longer smallpox in our environment. And hence, there is no children that are sick with smallpox post the vaccination campaign. And there are some more recent or more relevant examples of that too. So some data I provided here. This is American data, for which I apologize. But it's looking at the rate of measles. And the measles vaccine was developed in the early 1960s. And so you can see that the rate of measles pre-vaccine was about 1 in 200. 1 in 200 people will be infected with measles. Once the vaccine was brought in, you can see this rapid drop in infection rate. And so this is the impact of vaccines. The other half of this graph is quite interesting. So SSPE is an encephalitis that occurs as a rare consequence of measles. So in a few people who get measles, about 10 or 12 years later after that primary infection, they will present with a brain inflammation and it will cause severe brain damage, and that's SSPE. And the interesting thing is that once the drop in measles occurred, then 10 or 12 years later, you saw the same drop in this brain inflammation syndrome. And so measles vaccination has been incredibly effective until people have started refusing to take it. And so that's part of the discussion to have later. One of the questions then about vaccines is if they are so brilliant, what's next? What is current in the field? And I think there are a few areas that are really targeted by research at the moment. And the first one is that there is a good number of diseases that cause severe death and suffering that we don't yet have effective vaccines for. And there's a huge amount of research effort put into trying to design vaccines for these diseases. Some of them are listed here. So malaria is a classic example. Tuberculosis is another. HIV also. And part of the reason that we don't have vaccines for these diseases, even 40 or 50 years after the first vaccines were developed, is that these are particularly clever pathogens that have excellent ways of escaping the normal immune responses that we use. And so the vaccines that we need to design are more complex than the original round of vaccines that we use. So one is to tackle diseases that we don't yet have good vaccines for. The other research arm is to try and improve the vaccines that we do have. And so improvements might be reducing the number of shots that you need in order to get good protection against a particular disease. They might be in terms of the logistics, so designing vaccines that are easier to use, that are easier to transport to places that don't have a refrigerator storage or don't have easy access, or logistics in terms of the politics and the education required, or involved at least, around vaccine delivery programs. And so there's a lot of both science research and socioeconomic research going into vaccines at the moment. And so that was my introduction. Really what I wanted to say was the immune system can be an incredibly powerful thing. And that in terms of infection, we can use that in many ways, in both diagnosis, in treatment, and in preventing disease. So thanks very much.
Okay, thanks for the introduction, Nainan, and thanks for the terrific overview, Georgia, of the interworkings of the immune system. What I thought I was going to do today was to just focus on the area of tumor immunology in a nutshell. So to do that, what I thought I'd do is I would take you back to some of the basic principles, some of the discoveries made at the turn of the century, and then tell you about some of the new discoveries. Pardon me? The 20th century. Yeah.
19 to 20.
Yeah. And then move on to tell you, to provide a couple of examples of where the field is going. And then I'll end the talk with a short little vignette of my own research. Okay, so really the grandfather of immunology was this physician named William Coley. So he was a young bone cancer specialist who was inspired to improve cancer treatment following the death of his patient, Bessie Daschle, at the age of 19. So Bessie was one of his first patients. He was about 24 years old, and he was really inspired by her death and other patients' deaths to go back to the literature to try and find some better strategy for treating these patients. And so what he did was he reviewed a huge body of medical literature, and he came across a few really poorly described reports where patients had come into the clinic and had developed very severe infections. And miraculously, they had walked out of the clinic a couple of weeks later, often a month later, with no evidence of tumor. So he figured at the time that it was likely that the bacterial infection was releasing some sort of toxin that in an indirect way was having an activity on the tumor cells, killing them. And he first tested his ideas back in 1891 or thereabouts. Now, Coley's toxins can still be found today. They have not been approved by the FDA. They are not available for public use in Canada, but they are being used in China and in Germany in some cases. The data to suggest that they work by Coley's mechanism is very poor, however. However, even around that time, other researchers were postulating that it was going to be likely or that it was very plausible that the immune system would have the power to recognize cancer cells. Now, you heard in George's talk that pathogens are unique and different than self cells. Structurally, they're different. They have a repertoire of different cell surface components that allow the immune system to detect them in a very different manner than a host cell or host immune cell would, for example. But even back in 1909, Paul Ehrlich speculated that the immune system would recognize and eliminate developing tumors. This was followed up by other Nobel laureates hypothesizing and developing an immunosurveillance hypothesis. So Burnett, the famous Australian scientist, speculated that tumors may develop, and because of their possession of new antigenic potentialities, provoke an effective immunological reaction with regression of tumor and no hint of its existence. Okay, so that was in and around the 1950s. So those ideas made sense. We were becoming aware of the vast power of our immune systems. However, their ideas really languished. Over a period of about 50 years, there was really very little experimental evidence to suggest that the immune system really played any role in preventing cancer growth. However, things changed in the mid-90s. So some really seminal discoveries that occurred in the 70s and the mid-90s really illuminated some of the missing pieces that the immune system might need to generate a reaction against a cancer cell. So, for example, we found that the host immune system evolved a whole repertoire of receptors called pattern recognition receptors that had evolved to recognize particular moieties that were specific to pathogens. And those receptors basically provided the context for how the immune system was going to respond to the pathogen. And obviously, tumors lacked those sort of moieties. And so the hypothesis that tumor cells would be able to evoke a T cell response was less likely given that sort of information. At around the same time, a scientist named Ralph Steinman discovered the dendritic cell. Now, Georgia gave a very good introduction as to what the dendritic cell is. So this cell you can think of as a commander of the immune system. So they are present in very low frequencies within your tissues, but they have on their cell surface and intracellularly a vast repertoire of these receptors specific for microbial products. So they are the commanders, they are the generals, they're the ones that initially sense the invading microbe and basically call to arms those soldiers that Georgia talked about. So we now know, for example, that the developing dendritic cell has to undergo a profound morphological change for it to become activated. Those receptors that I talked about on the surface and intracellularly in these cells respond to the presence of pathogens, microbial products. And when they sense them, they're induced to mature, differentiate, and that process confers upon them an ability to activate those soldiers that we talked about. So the dendritic cells, for example, following that microbial sensing, now have the ability to engage T cells, different subsets of T cells in the immune response, both CD4s and CD8s. Cells that one would postulate would be capable of killing tumor cells if appropriately activated. Dendritic cells also talk to other cells in the innate immune system, activating cells such as cells called natural killer cells. So these discoveries suggested that there was a missing link in terms of our ideas of tumor immunology. It was likely that the immune system probably did a very poor job of immunosurveillance, the idea that your immune system would go out, seek and destroy, and kill small growing tumors. However, it suggested to us as researchers, now knowing what it took to induce the maturation and activation of these cells, that perhaps we could use that information to trick the immune system into recognizing tumor cells by providing these sort of pathogen-derived products to the tumor treatment regime. Okay, so what I'm going to do is I'm just going to, over the next few minutes, go over two cellular strategies that are now in the clinic to treat cancer. Okay, one of them is going to rely on that dendritic cell that we talked about, and the other one is going to employ the soldiers, those T cells that you heard about in the previous talk. Okay, so the general strategy, so this strategy has basically just been approved by the FDA for treatment of cancer patients. And what it entails is the drawing of blood from a patient with cancer the propagation of a precursor cell to those dendritic cells that I mentioned. You expand those cells in a dish in the laboratory, and then you can genetically alter those DCs to enhance their functionality, for example. You can feed them tumor cell lysates, apoptotic cells, recombinant proteins. Okay. And importantly now, we can provide a maturation stimulus. So knowing now what we know about that repertoire of receptors that dendritic cells express and the requirement for microbial stimulation to induce their maturation, we can add that to that culture system and then reinfuse them back into patients. Okay, so that's one of the strategies that has entered the clinic and is showing some promise. Okay. The second strategy revolves around using the soldiers of the immune system. So instead of trying to propagate those generals and to provoke them to activate T cells, this idea instead relies on us harvesting T cells from a patient. Now, in this case, the patient has to be the patient that has the cancer. Okay, so we harvest their T cells. We then genetically alter them. Okay, so we can infect them with viruses and within those T cells now express receptors that are specific for the tumors that we want to target. Okay, and that is done in a variety of different ways, and I'll introduce a few of those ideas to you in the next slides. So we increase their numbers dramatically in vitro. So whereas usually in your immune system you would require the general to talk to a few T cells, and then those T cells would be induced to proliferate to substantial numbers or enough numbers to tackle the infection. Here what we're doing is instead bypassing that first step, growing them in the lab, and infusing the patients with hundreds and millions of these cells, all of which are specific to the protein that we want to target on the tumor cell. Okay? So within about a 10 to 12 day timeline, the patient's blood is drawn, the T cells are enriched, they're genetically modified, they're expanded, and they're infused back into the patient. So providing a little more detail about those sort of strategies. So there are basically three ways that we can do this. One of them is we can identify basically tumor-infiltrating lymphocytes or T cells from the patient's blood. So if we can identify a T cell that's specific for the tumor and then expand it and infuse that back into the patient, that's one strategy. The other one is to genetically alter that T cell so that it has a receptor on its surface, a natural receptor on its surface specific for the tumor antigen. And the third strategy is using something called a chimeric antigen receptor. So in this strategy, what we've done is we've used pretty elaborate genetic engineering strategies. We have taken those T cells and we have infected them with a virus that expresses a receptor that does not exist in nature. Okay? It is a fusion of an antibody, which you heard about in the previous talk, fused to another protein that basically tells the T cell that you should grow, proliferate, and become activated when your receptor, that antibody on your surface, recognizes a specific protein on the tumor cell. Okay? So it's this strategy that in the last 12 to 18 months has really shaken the foundation of tumor therapy. Okay? This particular strategy is curing patients that had otherwise been on death's door. Okay, I'll give you a couple of examples of that. Okay, so in the case of B-cell malignancies, there are a couple of examples that are in clinical trials or have just completed their clinical trials. Two of them, chronic lymphoblastic leukemia and acute lymphoblastic leukemia. So in this particular approach where they took those chimeric antigen receptor transduced T cells and then infused them back into patients, 86% of children receiving those CAR T cells targeting this protein called CD19 This is a protein that is only expressed on B cells. Those patients showed complete responses in 19 of 22 of those patients. Some of these children had gone through 3 to 8 previous therapies, and 13 had had prior bone marrow transplantation. Basically, that was the therapy of last resort, and they had regressed after that. Similar efficacies are turning up in adults. So of the small group of adults that have been targeted, 14 of 16 adults showed complete responses after CAR T cell therapy. And in some of these patients, it's been estimated that between 3 and 7 pounds of tumor were killed within 7 to 14 days. So in one case, this particular gentleman was on a ventilator, basically near death, was infused with T cells, and within a month was back on the golf course. So this highlights a couple of things. One is that it's taken us over 100 years from some of these primary observations made by physicians, 100 years of basic science, basic research in developing our understanding of how the immune system works and functions. Probably 15 to 20 years of doing basic biochemical studies about how these receptors actually work and the signals that they send to make those chimeric antigen receptors actually work in patients. Of course, there's a downside to this sort of therapy. Some of the complications are that in this case, the T cells are targeting all of your B cells. So there's not a tumor-specific antigen now. This is a tissue-specific antigen. But because you can live without B cells, the idea is that you are better off to have removed this entire population of cells than to have died of your B cell cancer. The patients often develop something called tumor lysis syndrome, and that is basically a complication due to the fact that you have pounds and pounds of cells that are basically being popped open within you in a very short time frame. So you have to deal with all of those metabolites, all of that proteinaceous material, and it's extremely hard on your organs, your kidneys, for example. There's something called cytokine release syndrome that often develops in these patients as well. So if any of you have had a severe case of the flu, some of the ill effects of the virus are due to the cytokines that are produced in the ensuing immune reaction. The profound immune reaction that is occurring in these treated patients, the cytokine release syndrome can kill them in some cases, and there have been a couple of patients who actually died from the magnitude of the ensuing immune reaction. And similarly, there's something called a macrophage activation syndrome, which is a similar type of problem. There are also problems to think about in terms of how this particular type of science is going to survive in the clinic. So we're talking about between 50 and 100 patients so far. How do you scale this up to treat thousands, tens, hundreds of thousands of cancer patients with different types of cancers? Unlike traditional therapy, this sort of genetic engineering and cellular therapy requires more skilled personnel. You need skilled technicians that are able to handle patient populations of cells. You have to have much more sophisticated skill sets to deal with those retroviruses or those viruses that are used to introduce the genetic material into the T cells. So this is requiring a rethink about how this sort of strategy can be incorporated into modern medicine. In the States, this seems to be working relatively well in a pay-for-medicine sort of approach where some of these strategies are upwards of $200,000 to $300,000 per treatment. That is something that they have embraced. And there are companies scrambling to set up treatment centers all across the U.S. In Canada, we're going to have to think about how similar sort of small boutique type treatment centers could be set up. That being said, this entire program, this entire tumor treatment regime has come out of basic laboratory science. So these are investigators like us who are working at universities largely in medium-sized laboratories who have established some of these early trials. Okay, so the question is, how do we embrace this and move it forward? There's also the question of how we translate this sort of approach to more immunosuppressive type cancers. So in the case of a hematological or a blood-borne malignancy, the idea is relatively simple. However, for solid cancers, we know that a lot of them are profoundly immunosuppressive. So there's a chance that these sort of infused cells, once they reach the tumor, would become functionally inactivated. And that's actually a topic of my research program. Okay, so I've given you a couple of examples of how personalized medicine is moving in the immunological field. We are making use of those sort of early studies about dendritic cells, how to harness their functionalities to inject patients with the generals. And I talked about CAR T cells, how we can expand the soldiers and infuse those soldiers in patients in huge number to treat cancer patients. There are other strategies in the clinic as well, or emerging. Some of those include using monoclonal antibodies to reactivate exhausted T cells, for example. So often in cancer patients, you might find a small population of tumor-specific T cells. But functionally, they appear to be impaired or exhausted. So there are strategies now using those proteins called antibodies that you heard about to block those inhibitory receptors to reinvigorate those T cells. And at least 2 of those monoclonal antibodies are showing terrific results in the clinic. Okay. So my own work. So my lab is particularly interested in breast cancer. And we have found that breast cancer is one of a small group of solid cancers that has a number of tricks up its sleeve. So one of the things that it does, and it's particularly adept at, is it actually blocks the development of those generals. Okay, so those generals are all derived from a hemopoietic stem cell, a primitive blood cell in the bone marrow. They have to develop and then undergo that maturation process that I talked about. Well, breast cancer secretes proteins that effectively block that maturation process. Okay. So effectively, what that means is that a patient with metastatic breast cancer doesn't have the generals. The generals can't talk to the troops, can't generate those T cell responses effectively, and can't activate other arms of the innate immune system effectively as well. Okay, so we're thinking about ways to basically take apart that biochemical process to better understand what the secreted factor is and how they work on the developing dendritic cell so that we can come up with drugs to block those effects. Taking that one step further into the field of personalized medicine, what the laboratory wants to do is to move some of these studies using patient samples into better preclinical models. So, for example, we know that a large number of different solid cancers interact with the immune system and immune systems in very complicated ways. And I told you in this example, the developing cancer suppresses the development of the immune system. So how do you actually explore that in a laboratory? You can't really do those sort of studies in patients. So what we are developing is a humanized mouse model system where we take mice and we rebuild the human immune system within that mouse, transplant them with patients' tumors, and then that will basically allow us to have a research platform with which we can look for small molecules, strategies, adoptive cell therapy strategies to basically evoke more sustained and efficient immune responses against the tumors. Okay, so that's the dream. I'll just leave that, maybe leave this with one final thought, and that is that as a cancer researcher, we have to face the fact that since the war on cancer was initiated by the American government in 1971, if you look at the curve, if you look at the death rate from cancer and heart disease among women and men younger than 85 years, it's a pretty depressing line. So whereas changes to behavior, diet, some really impressive drugs have helped us treat heart disease, cancer has been really an immovable object. So the hope is, though, that now with these immune system approaches that we'll make a dent and send that curve lower. So, okay, I'll end there.
Need that snake venom thing. Okay, so thank you everybody for coming tonight, and it's really difficult to follow two great talks, but I'll give it a go. Hopefully I won't put you to sleep. How many of you know about or know someone that has had multiple sclerosis, type 1 diabetes, rheumatoid arthritis, lupus, all of them? All right, so all of these diseases are quite different. MS affects the brain, diabetes the pancreas, lupus is systemic, arthritis is our joints. There's hundreds of autoimmune diseases, and they all affect different parts of our body. And yet, somehow, in 10 minutes, I'm supposed to tell you everything about the immunotherapies of autoimmune diseases. All right, so let's just define it so we're all on the same page. Autoimmunity is actually nothing more than simply our body's immune system reacting to ourself, okay? And autoimmune disease is that rare opportunity when that autoimmunity, which we all have, actually causes disease. And it's really a small subset of us as people that unfortunately get those kinds of things. And so, unfortunately. Autoimmunity is not uncommon. We all actually generate, and I'll show you how that happens. It's natural for us to have it. It doesn't typically cause us any harm, and it's only in the minority, and some people are here, that it does cause a problem. But in all those hundreds of diseases, the etiology is all the same, okay? And that's what's important and what I want to show you tonight. Because when we think about therapy, it's common for all these diseases. And it's really, the cause of disease is similar. It's the location and the clinical manifestation that are different. MS is different than diabetes. There's patients out in the world who will have one autoimmune disease, and their relatives will actually have a different one, but they both have autoimmune diseases. Okay? And so it's not a single cause of autoimmunity that happens. It's actually very multifactorial, but we can break it down into two things, genetics and environment. And there's many factors in each that cause these things. So it's quite complex. And so the diseases themselves, we can divide them into two categories, systemic and organ-specific. Type 1 diabetes, the organ-specific ones are a little bit easier to wrap our brains around because we're involved with a particular organ, and we can think about it as an immune attack on that organ. So in type 1 diabetes, which is an example of one of the diseases that I use in my research and I'll use tonight, is an attack on the pancreas. It's an attack on the beta cells in the pancreas that make insulin. And multiple sclerosis is not only an attack on the brain, it's an attack on the oligodendrocytes that line the neurons and give them connectivity to talk to each other. Autoimmune myocarditis involves the heart and can cause scarring in the heart, which will lead to heart attacks. Myasthenia gravis causes it's problems with our neuromuscular junctions, and our nerves and our muscles can't talk to each other. Hashimoto's thyroiditis, which I have two eyes in, is actually a disease of the thyroid, or it's attacked by the autoimmune response. And Addison's is the adrenal glands. And not to go through it all, but lupus is a systemic attack against a number of cells throughout our body, not in an organ-specific manner. And arthritis is really the joints, which we have throughout our body, or our skin, that we get these attacks. So in MS, and I know this is probably a little bit blurry, this is a patient that has MS, and this is MRIs 3 times in their life. And you can see these large lesions or plaques that form where the autoimmune cells are going in and attacking and destroying the oligodendrocytes and the myelin. And this is an example of diabetes, where you start off with an islet which has virtually no immune cells. And Beta cells make up over about 70% of these cells. And slowly these little blue dots, blue is bad in these things if you ever watch them, blue are circling the islet and eventually coming in over time and destroying the islet. But the interesting thing is here, you need to destroy 75% of your islets before you even know that you have diabetes, before you can see that you have high blood sugar. And I'm just going to go back a second. If this lesion isn't in an important area, you don't even know that you have it. Okay? We only use about 10% of our brain. So if these lesions don't pop up in a spot that we need, we won't know that we have it. And when they do pop up, in every person that has MS, it's slightly different. And this is a key important thing, because when we think about autoimmune disease, and this example is diabetes, you're trucking along, as a colleague of mine calls it a train, but you're trucking along on a train with your genetic predisposition until you hit an environmental event that triggers it off. And then you get worse and worse and worse and worse, and still nobody knows. But eventually, you run out of islet cells, or you're down to your last 25%, and now you have overt diabetes, and it's too late. We've actually lost all this tissue. And depending on the autoimmune disease, that's hard to get back, okay? And to get a good therapy, we really need to do things back here when we don't know that you have it or that you'll even get it. And so that's the difficulty with autoimmunity. And so, just to say that point in a different way, tons of people are genetically susceptible to getting autoimmune disease, but it's only a minority of them that actually get autoimmune disease. All right? And so it's a difficult, complex disease, but it's very easy to get autoimmunity. And this slide is based on a colleague of mine and my work with Coxsackie virus and how it can induce autoimmunity, which it does in people and it's actually a human virus, but we use it in mice and can model it. But the virus just simply infects a target cell, which can be a pancreatic beta cell that makes insulin, or it can be a cardiac myocyte. And it causes an immune response. This is natural. This is needed. That T cell response will then go right to the virus, attack it, clear it, get it out of the system. It's fine. But while it was attacking these cells and identifying them, it identified antigen or proteins that were in this target cell that are our own, that are self. And it decided, well, we need to make T cells to everything we see, and we did that, and we get this huge T cell response and B cell response and antibody response that all goes back and attacks target cells that are no longer infected, causing autoimmunity, and in this case, potentially causing diabetes. And so this is a question of, Georgia brought up, there's an army, and other people call them a police force, but we need internal affairs here, right? We need people to be watching these cops that are actually killing these target cells, because we have to tell them not to do it, right? Because it's bystanders that are dying here, okay? And that's what autoimmunity is about. And so how do all of us who get autoimmunity regulate autoimmunity? Well, that was summed up in the last two talks quite a bit, but Coxsackie virus in itself causes a number of T cells that are very strong and effectively, and they're called T effectors because they effectively remove the virus. Unfortunately, they also effectively remove some of the pancreatic beta cells. Okay? But for every T effector that's made, we need to also make regulatory T cells. And when we get infected with a virus, what's interesting is we want a lot of these effector T cells, but we actually want to calm down those regulatory cells. And we do that by making what's called interferon. Okay? And interferon will upregulate all our T regulatory cells, and it downregulates, or it uprates the effectors, downregulates the regulatory cells, and that's just for a short time. And then interferon will be gone, and those numbers are supposed to balance back up. Okay? So if we have a lot of regulatory cells to monitor our effectors, we get no disease, and we remove the virus. But if we can't get those regulatory cells on track, that's when we get autoimmunity. And what controls that are cytokines, which were introduced previously, and more importantly, our genetics. Our genetics tells us how to control these. Okay? And those genes are where the susceptibility genes are usually located. Okay. So when we treat autoimmunity, we don't want to turn it all the way off, all right? Because that will, of course, get rid of the autoimmunity, but we'll have no immune defense against any viruses, bacteria, or cancer cells. Okay? And the problem is that most treatments that we have out there, this is what they do. And so they try to give it to you for a short period of time and then take you off and try to ease you back and forth on it, right? But this is not necessarily effective because the autoimmunity just comes back. And so if we don't do anything about it, we have to live with our autoimmunity, but we have a healthy immune defense for a while until we can't take it anymore. So we really need this balance between there, somewhere where we can put it in between. And so, like I said, historically, the drugs that were used to fight autoimmunity are the corticosteroids. Prednisone is a great example. Most of you have heard of prednisone and the ills of prednisone. But if you give prednisone, your autoimmunity will stop dead in its tracks. Of course, so will your defense against viruses, bacteria, and cancer. Okay? And so that's the main problem. And so many researchers, I'm going to sum it up in one slide, have done a tremendous amount of work identifying all kinds of therapies, okay, to attack all these cells and keep them into control. And so just because you can't probably read it all here, these are B cells, which can also act as antigen-presenting cells. And rituximab is a drug that's been developed to downregulate B cells, and it works very well in MS patients for a while. Okay? There's T cells. This is a naive T cell before it's activated. And there's drugs that can affect naive T cells to keep them modulated. There's effector T cells that are activated. In this case, it looks like it's getting ready to kill a pancreatic beta cell, or at least identify one. And there's drugs all over the place that try to teach these guys, one, not to attack through what's called oral tolerance. But there's other drugs like anti-IL-1 beta, which will try to downregulate macrophages, which are activating the whole situation. But there's tons of trials, and many of these are in clinical trials now with mixed results. And as we learn more about the mechanism and the etiology of autoimmune disease, we identify more of these potential therapies. And there's just too many to list, and I don't want to bore you to death, but if you want to talk to them about me later, you can. But what we did notice in the world today, most autoimmune diseases, especially type 1 diabetes and multiple sclerosis, are first world diseases. Okay? They're not found in the third world at any high level. Okay? And what is found in the third world at a very high level is, unfortunately, a lot of other pathogens. Okay? Or it's not so hygienic. And so it came up with the hygiene hypothesis. All right? And the thing about the hygiene hypothesis is that part of it makes you believe that there's something out there, some pathogens out there, that can actually maybe protect us from these ills of autoimmunity. Many of them probably don't help us live our daily life past 3, but many of them probably do help us. And so people have looked at that, and they found that a lot of people in third world nations carry a lot more worms in their intestines than other people. And so there's researchers that have gone and gotten helminths, and I don't know how they got them out of there. I think it's really cute, though. And they've given them to mice that get type 1 diabetes or a version of multiple sclerosis, and they find that the helminths work. And when they look at the immune system, what the helminth does, just by being in the gut of these mice, is to take what would normally be an autoimmune situation with all these bad T cells and dendritic cells and macrophages, and with few or non-functional regulatory cells going on, is it evens out the balance. Okay? We don't exactly know why it does, but one reason is the worm is trying to survive. And for the worm to survive, it needs the immune system like this. Okay? And by doing that, by distracting the immune system in a very major way, it really corrects for these ills. Because the reality is that the immune system really doesn't want to, if it has wants, it doesn't want to attack us, right? And when it does attack us in multiple sclerosis and type 1 diabetes, it's not like how it attacks influenza. When it attacks influenza, it does it the next day, immediately, and it removes as many cells as possible. With MS, it's a period of time. It takes a long time to get out all those oligodendrocytes that they really do want to attack. So there are controls that are working. They're just not working to their most effectiveness in autoimmune disease. Okay? And so it's my opinion that treatments like this that are more systemic and more universal, like the helminth treatments, and maybe other viruses they're part of our microbiome or virome or bacteria, are really going to lead to these, ultimately finding out what the controls we need to change these things are. And so pretty much that's the state of the art today. I welcome any questions and everything, and thank my colleagues. They did a great job.
Okay, so I'll ask each of the panelists to come up as I raise a question, and we'll speak into this mic so everybody can hear us, and we'll be recorded. So the first question was a really basic one, which I'm going to have Georgia answer because it's actually both our research area. What is a cytokine? How does it work? And or what is its purpose? No, please come forward. I'm just moderating today.
I've forgotten the question already. What is a cytokine? Let me do that first. So a cytokine is one. Get close to the microphone. A cytokine is just one of the many molecules of the immune system, but it's one of the key communication molecules. So it's something that one cell makes, and it's a molecule that's secreted by that cell, and it acts on a target cell. And that process of being secreted and being received by a target cell is a process or an exchange of information. So you can think of a cytokine as a set of instructions, and the outcome of that exchange of information is that the target cell changes in some way. Its behavior or its function changes in some way. And there's a whole list of cytokines. We tend to terrorize our undergraduate students by asking them to remember what each one does. There are lots of them, and they do very many things, but they keep the immune system working as a coordinated beast.
Great. You answered all
Is
the
that
questions.
excellent?
So she addressed how it worked and what its purpose was. Oh, absolutely. She's a brilliant speaker. Brilliant speaker. I never hope to follow her. Okay. Next question. I think, Ken, you might want to address this one. How accurately can a person's immune system state be measured? I want to touch on the mass spectrometry changes.
That's actually a hard question to answer. I mean, certainly if you go into the doctor's office, we use very crude approaches to assess it. You know, you'll have your blood drawn. It'll be run through a machine, and we'll be able to tell you the breakdown of the composition of your blood. But that actually provides very little information about the functional competency of any of the cells that we're looking at. The only thing I'll add to that is that some of us, myself included, are working with a new technology. It's a very expensive contraption that will allow us to, using mass spectrometry, which I'm not going to get into because I wouldn't be able to get into it, but it's a machine basically that will allow us to measure somewhere around 100 parameters on any given cell. So we can look inside the cell to see whether that cell is activated, whether it's producing those carrier pigeons or cytokines that Georgia talked about, what the cell surface expression profile is of all of the receptors that are on that cell that tell us about what it does and how it functions. So, Mark, do you want to help me out on this? Is there anything else? Yeah.
So the question's, how can you tell if what your immune state?
Yeah, the immune state.
Well, just depends. So there's so many things that the immune system does and functions to do. And one of the ones that's really easy is that, of course, you go to the doctor and they look at how many white blood cells do you have. That's an easy one. Some of us have over the normal and some of us lower. I mean, if you have lower, that's not good. And if you have too many, that means something's happening. So that's just a very simple one. The other one, of course, is looking for antibodies that are responsive to any virus that you may have had, which is good to know if you've been vaccinated or you knew that you had chicken pox. It's nice to know whether you still have antibodies. If there's a reduction in that level, then they could tell that there's something going on to diminish your abilities to respond. And actually, they do this, I think, with geriatric patients. We look at the T cells and the antibodies that are specific that you have, because as you get older, more of your antibodies and T cells are directed towards a virus called cytomegalovirus, which most of us carry in our bodies, and it tends to push all the other immune responses out.
What is the cytomegalovirus?
Cytomegalovirus is a virus that is really commensal to all of us, and we learned a great deal about, unfortunately, during the AIDS, because of AIDS. It's still an epidemic. Because if you have AIDS, cytomegalovirus will give you pneumonia. And so, but most of us, something like 75 to 80% of us as adults, carry cytomegalovirus. It's a virus that we get infected with, and then we carry with us for the rest of our lives. It's dormant, relatively dormant, unless there's an immune or environmental event that stresses it out and it reactivates. but like getting AIDS. But over time, our immune response to cytomegalovirus just grows and grows and grows. And that's part of the process of how it stays dormant and how we keep it dormant. And by doing that, it kind of excludes out all the other responses we may have to chicken pox or whatever we were infected, influenza. Yeah. Does that help?
That helps. Thanks. So just to add to that, HIV is actually informative because it's a virus that infects the immune cell type called CD4. So when a doctor sees someone with multiple different diseases cropping up in that individual, the simple blood test is informative. You can measure CD4 cell counts just doing a simple blood draw. But most of our immune disorders are much more complicated than that. And the person I'd refer, whoever asked this question, Mark Davis is a researcher in Stanford using that technology that Ken alluded to, the mass cytometry method. We will now get baseline reads that are far more informative. It's going to be big data, the data cloud, new word I learned today. That data cloud will include our immune parameters, and it's just a matter of funding those tests. Okay, next question. How do T cells differentiate tumor cells from other cells? Who wants to take this? Georgia? You want to take this? No? Ken? All right, Ken. You're the tumor person. And then I've got follow-up.
Okay. The simple answer is that tumors develop due to acquiring mutations in their genes. And those mutations introduce mutations in protein sequences, and those protein sequences, all of the protein sequences that you have in your body, are presented on the surface of cells by way of a receptor called MHC. And so if your tumor cell has a mutated gene within it and makes a mutated protein, small pieces of that protein will be presented on the surface of that cell, and your T cells will be able to recognize it as being different. Okay? I'm not going to go into T cell tolerance and T cell development, but suffice it to say that the normal protein component of your body, you have educated your T cell population such that they ignore self-protein. Tumor proteins are different. Okay? They're mutated self. So your immune system has the capacity to discriminate them. Okay? Is that clear?
The follow-up question was, have there been long-term effects of treatments like B-cell malignancy for CLL and ALL? Long-term effects of treatments. Yeah. So in CLL and ALL, what are the long-term treatment effects? And what's being done to catch autoimmune disorders early and prevent them altogether? That's from Mark.
Okay. So what was the question?
What are the side effects of treatments?
So side effects of these new radical approaches, I listed a few of them, some of the acute toxicities. Long-term effects, we really don't know. Okay? These are cutting-edge strategies. Some of the patients who have gone through the first trials are still alive 3 to 4 years on without any adverse effects, other than not having any B cells. Okay? But there are treatments that you can get to replace your immunoglobulin that all those patients get. One of the real worries about this strategy has been that we have to rely on using viruses to introduce that genetic material into these T cells. And there's been worries that those viruses could integrate into our genome in a way that would unleash cancer-promoting genes. Okay? That doesn't seem to be true. So there is now enough evidence, historical evidence, using those same sort of viruses in trials of patients with HIV, where we tried to introduce new types of genes within their blood system, that we can now track those patients out 20, 25 years, and there's no evidence that those viruses cause any ill effects.
The third part of this question was, what's being done to catch autoimmune disorders early and prevent them altogether?
Well, we know a lot of the genes that allow for susceptibility to getting autoimmunity. And because of that, we know, at least in a familial situation, a family, what's going on. And so what people are trying to do is track those individuals. And so if a member of your family has, for instance, type 1 diabetes, and the hospitals and centers around in North America try to do a good diligence of getting all the family members to come in and try to follow up with them over time. And with type 1 diabetes, it's generally a disease of children that is when they start to get it. So usually all the other children in the family. And then they try to follow up and watch them for early stage markers, but also suggest that they join what's called TrialNet to start some of these treatments early before the onset of type 1 diabetes to try to stop it. So the hardship, of course, that is that for a patient is that you're looking at a situation where you are not sick, or your child's not sick, your other child is sick, and you're thinking, maybe I should go ahead and start this treatment or not. It's very much how many of us started taking lipid drugs. The doctor said, your cholesterol's high, right? So you got to take that. But you're not feeling ill, but it's just a number that's being thrown at you. So compliance is not always there. But the thing that we don't know is what causes the diabetes or the multiple sclerosis or whatever the disease is in the patient that's genetically susceptible. And for all these diseases, we have a very small idea of what some of these causative agents are. And there's a lot of people out there working on that. So until we know that, we really can't.
Being environmental.
We believe very strongly that it's both. You have to be genetically susceptible. You have to have a genetic component. But there's a very strong environmental component, and that component is likely comprised of multiple events, not just a single event. And that's the rarity of the disease, because we know that in twin studies where they have the exact same genetics, that differences in environment make all the world of difference in terms of getting disease.
That's actually a great segue. There are actually two related questions, probably for you, Mark. What role does stress and diet play in autoimmune diseases? And the other question was, what about exercise, lifestyle, food we eat as immune system boosters, and therefore as preventative medicine for cancer and many other diseases? You want to take that?
Well, stress and diet, I don't have autoimmune disease, but just say that. But there are probably big factors in other things. So for autoimmune disease, stress and diet probably have the only factor they have is as an environmental component, obviously, and how you handle that. So I don't think there's a lot of evidence that says that stress and diet are important, other than the fact that they relate to the level of vitamin D that you might have in your body, or how the types of vitamins that you're eating. They're all going to be part of the componentry of getting a good control and regulation of your immune response. Or on the other hand, just a healthy immune response. Diet and stress are very related. So, but I don't think that they're actually going to be the event that we're talking about that precipitates disease. And so the second part was,
What about exercise, lifestyle, food, in immune system boosting?
all of that's pretty similar. I think that's the same real answer. Right? It all helps. It helps to have a good diet. It helps to have good lifestyle, good exercise. But if it's a virus that causes type 1 diabetes and it's occurring in a 1 or a 2-year-old, is when they're starting to get this. That I think all those things kind of go out the window. But they are important in keeping that disease down and under control.
One of the microbiome experts in the room want to weigh in here, Ken? I know Pauline Johnson's in the room. She's also an immunologist, but she's ducked out. Is she hiding? Okay, Ken, why don't you address the role of the microbiome in immune wellness?
Okay. So basically what 9N is bringing up is this idea that the constituents of our gut, the microbial constituents of our gut, play an important part in our predisposition to disease, to chronic inflammatory diseases like inflammatory bowel diseases, to colorectal carcinoma, for example. And so there is emerging evidence that dysbiosis, this idea that your microbial populations in your gut may shift to allow populations of bacteria that negatively impact your health, I think is now firmly footed in reality. There is really good evidence that these sort of shifts in micropopulations in the gut can predispose you to developing colorectal carcinoma. And there are obviously, I think, connected dietary issues to that. The fact that particular diets may do a better job of nurturing particular classes of microbes, which may themselves be leading to some sort of underlying baseline inflammation in the gut, which precipitates colon cancer, I think is now firmly established as well. So what does that mean in terms of what we can do about it? We know very little about those complex communities and how to shift them, despite the fact the fact that Activia would tell you that they know everything about how to do it. We actually don't know a whole lot about it, but it's a real active area of investigation. Sort of this idea of personalized medicine. In the future, you will have your genome decoded on a chip. You will bring that to your doctor, and likely you will have the composition of your intestinal tract decoded as well. So we will know the communities that live in your gut. We will know your genome, and we will hopefully be able to say something about how those two may interact. If you have a particular population of bacteria in your gut and you have a variety of polymorphisms in genes that are associated with propensity to developing inflammatory bowel disease, your physician may suggest strategies to alter that composition of the bacterial populations in your gut as a strategy. But that's science fiction right now. We're not there yet, but that's where we want to go.
Little known fact, the number of bacteria in your gut, 100 trillion, outnumbers your human body cells by 100 to 1. So we are more bacterial and viral probably than we are human in terms of cell numbers. So that's a little known fact, but that's how much we've co-evolved with our microbiome. So along those lines, someone's asked a question which I actually have never heard of. Have you heard of bacteriophage therapy? They're good viruses that are better at fighting antibiotic-resistant infections. And so the comment was there is a phage therapy center in Chibili, Georgia, which I assume is in Georgia. Anyone heard of bacteriophage therapy?
I will just say that I have heard of them. In the Soviet Union, or the old Soviet Union, bacteriophage therapy and research divisions were extremely popular. It is a proven therapeutic strategy for ear infections, for example. I gather, I'm not sure that this is true, but I had heard that if your child goes into a health practitioner in the Soviet Union, you are more likely to get a bacteriophage injection in your ear than you are to go on oral antibiotics, or at least you were. So it's a good question and something that we don't often think about, but promising avenue of research.
We talked about the farther rather than the nearer in Georgia.
Yes.
Correct. Indeed, in Russia, right. Georgia, this is your question because you introduced the concept of barriers. The second part of the same question was, is it better to swallow our saliva than to spit it out? It gets worse. We'll get out of the gutter now. Does our saliva strengthen our immune system? And then what about I'll leave it as nasal mucus. What about swallowing your nasal mucus? It says snot, but I was going to avoid that.
I said, is there a benefit to picking it and eating it? Is that your question?
I have kids.
I don't want to go there. So some of the immune defenses in our saliva are there for a reason. Like, they're actually useful in our mouths. They're the very first defense that you need. So there are enzymes that will break down bacterial cell walls, for instance. And so they're possibly more useful in our mouths than they are in our guts. If you swallow the saliva, well, you do. I mean, every time you swallow any piece of food, you're going to swallow your saliva, right? But anything that you swallow is going to end up pretty soon in your stomach, and the stomach is amazingly acid. That's important for nutrition, but that's also an immune defense. So the pH in your stomach is particularly good at killing off bacteria too. So there are different defenses for different places in our bodies, and there's a reason why they are there in the right place. In your nose, there are a number of things, reasons why you have mucus there. There's a number of reasons why you have hairs in your nose too. And so the fact that it is sticky is useful, because the hope is that if you breathe in bacteria, it will actually just get lodged in that mucus there, and hence blowing it out is useful, because you want to get rid of that bacteria, not to take it anywhere else into your body.
I would add that we evolved to swallow that nasal mucus, and it gets, whatever bacteria is in there gets destroyed by the pH in your stomach. So we co-evolved to do just that. No reason not to. Okay, Ken, for solid tumors, how can we get the designer treatments into the tumor, i.e. not just on the surface of the tumor?
Yeah, that's a really good question. The whole problem with solid cancers is one that I think is going to be a little bit of a roadblock for some of these therapies. That being said, tumors need nutrients. They need a blood supply. They're often highly vasculature, often leaky. So I think it is quite likely that these sort of T cells would get into the tumor. They may require a little bit of provoking to get there, though. And one of the strategies that's being looked at is where I said that you could genetically engineer those T cells to express a receptor specific for a tumor antigen. Well, what you can also do is you can put in a receptor within those T cells so that they are targeted to the tumor themselves. So the tumors actually will release these small molecules like cytokines or things called chemokines. And we can biopsy those tumors and find out what sort of chemokines they secrete, and then put the corresponding receptor into the T cell so that that T cell will naturally now home into the tumor environment. So another strategy to help those T cells get into the tumor is by local radiotherapy. So by inducing inflammation and cell death at the site, we induce inflammation, and that actually helps to bring T cells and other immune cells into the tumor as well. So there are a lot of different strategies being looked at to get those cells into solid cancers. And when I say solid, I don't mean impervious. Okay? They are still highly vasculature, and often those particular solid cancers, for some of them, up to 30 to 40% of the mass can often be immune cells themselves. Okay, so some immune cells are already there. They're just not the right type of immune cells.
Same related issue. Ken, how do you work out which antigens are on the tumor cells but not on other cells? Are there known antigens for particular cancers, or are they specific to each patient?
Yeah, that is an extremely hot area of research right now. We have identified a large number of tumor antigens. In breast cancer, for example, somewhere between 16 and 30% of breast cancers overexpress this antigen called NU. It's a receptor that's expressed on those tumor cells. In the case of brain cancer, in the case of prostate cancer, there's another protein called mesothelin, which we know is expressed by those cells, which we can use T cells to target. Where the field is going, again, goes back to this personalized medicine approach. With the very near future $1,000 personal genome, the idea will be that you will have your personal genome known, and a tumor biopsy will be able to be taken, and we'll be able to sequence your entire genome of that tumor, identify the nature of your mutations within that tumor, construct specific peptides or proteins that encode those mutated antigens, deliver them to those generals that I talked about, and then infuse them back into you. So that is where the future is. The future is going to be using these genomic approaches, genetic approaches, to basically catalog your entire mutanome, if you will, compare it to normal, and then use that information to generate specific immune reactions to your tumor. Well, you can have your entire fecal microbiome sequenced by a company for about $100 right now. In the case of your personal genome, it's somewhere around $1,000 to $2,000. So you can send samples away, cheek swabs. Tumor biopsy, you're going to have buy-in from a clinician. But I would say the $1,000 genome is probably here, almost. It's almost here. I would say 6 months. You know, in terms of accuracy, getting to an accuracy where we would want to use the information from that tumor biopsy to accurately say that those are real mutations and not sequencing errors, I'd say a couple of years. So.
Our genome sciences connection there is flashing 5 years, so that's about right, I'd say. 5 minutes? Oh. Well, we have 5 years here. Okay. So this question would probably be best addressed by Mark, who's in the room, but if it doesn't get addressed by Mark, our colleague Francois Jean works in hepatitis C. So the question is, what are your comments about new era antivirals for hepatitis C? And examples, sofosbuvir and semaprevir, 96% successful even in most difficult genotypes, but in small studies so far. Ever heard of these?
Just now. No, so I can't answer that question directly there. Was Francois here?
No, it's not.
Oh, okay.
So I would...
Have you?
Well, I don't really know much more than Matt does, but I'll keep talking anyway. So I don't know how these new drugs work. I know that they were released within the last year. They were approved by the FDA in America for use. They are amazingly successful, as the questioner knows. The catch with them that they're also amazingly expensive. I can't remember exactly what the dollar amount was, but somewhere in the region of $80,000 for the initial course. And so there are issues to deal with, actually, as Ken referred to, in terms of this might be a treatment that is supported by an American system, a health insurance system, and is less supported by a Canadian system. I don't actually know the details of whether they're being prescribed here or not. But it was an amazing breakthrough in hepatitis C research, that for a long period we had no way of treating the very resistant patients. So we have interferon therapy. There are groups of patients that doesn't work in, and this was an amazing breakthrough that would offer hope even to these patients. Unfortunately, I don't know more about how they're actually working. I'm sorry.
Great. If you have further questions, Francois Jean, Microbiology and Immunology Department, UBC. You can email him. He might be able to direct you. Okay, final question. What do you think of Jonathan Schneck's, Hawkins University researcher, work on artificial antigen-presenting cells? Is this a viable strategy for cancer or infectious disease therapy? Ken or Georgia? Artificial APCs?
I will first off say that I don't know about this researcher's specific work, but I do know a lot about artificial APCs, and they do show a lot of promise. The idea there is that you could use small nanoparticles or chemical structures to immobilize specific antibodies or ligands onto the surface, so you don't have to use a real cell. So you could take a tumor, a mutated tumor antigen, for example, emulsify it in this concoction with specific agonists of your immune system, and then infuse that to get around having to use a real cell. My personal bias, however, is that I like the idea of using a real cell to do this. You know, I understand that having an off-the-shelf sort of moiety that you could just put into patients is good, but having cells that you can actually derive from a patient, transfer back into them, and those cells can expand and contract. In the case of those T cells I talked about, you have a drug basically that will live within you for the rest of your life. So T cells will become memory T cells. If your tumor comes back, those T cells will expand, and they will kill your tumor, and then they will contract again. So I personally like the idea of cellular therapy over these sort of artificial antigen-presenting cells.
I can add to that. In terms of autoimmune disease, this has been worked on for a long time, and not necessarily with artificial APCs, but real APCs. And what they do is they treat the APCs so that the proteins are fixed on them. So what happens is the APCs like to engage the T cells and turn them on. But when their proteins are fixed, it only engages signal 1. So it recognizes, but the second signal is missing. And when the second signal is missing, that actually instructs the T cell to be energized or not to react. Okay? It also helps to develop regulatory T cells. And so for a long time, people are trying to use APCs that are coded or treated as possible treatments to reduce the effect of basically autoimmune T cells or T cells that are going to attack you. And so that's been a long therapy. It's actually currently all kinds of caveats with that, with nanoparticles and stuff right now are being used, as well as gel matrices on top of the molecules. So you don't need to be artificial. You can use real ones.
So just to put those in terms of George's analogy, the antigen-presenting cell is that general cell, the dendritic cell. That's a generalization, but you could go with that. So that's what we were talking about, engineering those dendritic cells or some other cell types that are like that to do this artificially. Okay, so we've reached the 2-hour point. We're all done here. Before I go, I hope this was as informative for you as it was fun for us to bring to the Vancouver Public Library. I'd like to acknowledge the Life Sciences Institute at UBC for its support in bringing this, and it was great that we got to be the first ones to do our VPL public talk of the LSI researchers. I don't know if there's anything back there for the public to take with them, contact sheets or anything like that, but there's an information table. If you would like to browse whatever's there, feel free to do that. Thank you, and have a good evening.